A series of non-P450 webinars. I started maybe a year and a half ago, maybe 2 years. And we started with things like UGTs, right? So the ones that most commonly encountered. Machi, who will speak later, focused on sulfotransferases. We had a couple other webinars, and it became pretty popular. We found, a lot of companies, there's been this trend to try to move away from P450s to some extent. In some cases it's successful, others not. But I'm finding kind of a resurgence in interest in carboxylesterase 1 and 2, which surprised me a little bit. And I've heard some more of that even just this week. I've gone around Scandinavia and given this presentation to different customers. And then the quick disclaimer, that these are my views, not necessarily bio-IVTs, especially any jokes that may be bad or good or what indifferent. And so basically I'm just going to go through an introduction, where the localization is of these enzymes, which is important, and introduces some species differences as well. And then I think what are really truly opportunities with carboxylesterase, the prodrug strategy has been around for years, but there are other newer ones I'll talk about. And then challenges, I mentioned species differences, I'll cover that again. And then simply just the regulation of these enzymes. There's, for the most part, only rodent studies, but I'll cover that a little bit. And then drug-drug interactions with carboxylesterases and some of their importance, and even at least one polymorphism. And then how to de-risk. So if you have a compound that looks like it has one of these functional groups, whether it's an ester, amide, thioester, whatever, how you would look at the victim and perpetrator potential. And I should mention, actually, I was talking to someone about this earlier. So the ICH has had this M12 DDI guidance out there in draft for about 2 years now. And FDA and Health Canada met in February, and they're announcing that they anticipate that to be finalized and adopted. They said in March, but I haven't seen it yet, so I'm guessing they're getting very, very close. And one of the things that they did change was they're no longer going to call it victim and perpetrator, they're going to call it object and precipitant. It was one of the 1,000 comments that they received that this was potentially offensive. So I think it's just a more scientific terminology. I'm kind of glad to see that. So if you look at the serine hydrolases, and they have this common motif, so you've got the regulatory domain, then the alpha-beta domain, as well as the catalytic triad. So these all are in common. There's, like I mentioned, several of these across different families, but carboxylesterases in particular are interesting in that in human there's essentially only 1 and 2 enzymes. That are involved in basically xenobiotic metabolism. And they catalyze the cleavage of ester, amide, thioester, or even carbamate containing compounds, as well as some others. And then, finally, deciphering, basically, depending on the structure, this is now one of the enzymes, carboxylesterase 1 and 2, that the guidance documents have added. Like if you don't have P450 involved, looking at non-P450 enzymes, and before it was sort of, maybe consider it, now it is definitely listed as one of the enzyme systems to consider going forward. So cellular localization, so this is important, again, for species differences, but if you look at this KDL receptor that is present in most species, has a number of different functions, but one of the things that it does is it will, depending on the C-terminal of the enzyme, for soluble enzymes, will actually sequester those in the endoplasmic reticulum. And so in humans, you have this HXEL sequence, and that retains carboxylesterase 1 and 2 in the endoplasmic reticulum. Now, it's not perfect because there are actually still a soluble fraction in the cytoplasm. And so you can see some differences in some cases where you might have some carboxylesterase activity in the cytoplasm. But this gets to one of the major species differences where in rats and mice, they have a different sequence. And because of that, especially for the CES-1 enzymes, they are actually secreted into plasma. So they're cytoplasmic, and then they actually also end up in plasma as well. So if you look at tissue distribution, so this is the Human Protein Atlas. I like to use this website because it typically has these mRNA profiles, but it also will have the proteomics data when it's available, along with some tissue slices showing localization. So this is a very useful website if you haven't come across it before. But you can see from this, carboxylesterase 1 is liver and then gallbladder. There's really not much else going on there with the mRNA. Carboxylesterase 2, on the other hand, it is still in liver. You can see quite a significant amount, and then these are different portions of the GI tract. So we think of carboxylesterase 1 as being the predominant liver carboxylesterase, and then carboxylesterase 2 is the one that's in the gut. And then if you look at proteomics data, so these are data that actually came out of the Prince Consortium, which our own Scott Hayward was involved in, I think, as a co-author on this paper. And you can see here that carboxylesterase 1 lights up very, very high proportion of the protein in liver versus carboxylesterase 2, which is still significant. And then moving to the intestine, you can see quite a bit more carboxylesterase 2, as I mentioned, in the intestine. There's still a small amount of carboxylesterase 1, and it's also present in other tissues like kidney, lung, and heart. So there is a good correlation, though, between the mRNA and protein expression. And then this graph was one that really surprised me when I found it. So this is, again, a proteomics approach. And what this is looking at solely drug metabolizing enzymes that are present in the endoplasmic reticulum and the amount of protein that's present. So, we're used to looking at this pie without carboxylesterase, and we're looking at P450s versus UDTs. Obviously 3 and 4 is a large one, and then some of the other UGTs. But carboxylesterase, by this study, is 43% of the protein that is a drug metabolizing enzyme in the endoplasmic reticulum. So a very high proportion of carboxylesterase 1 in human liver microsomes, and then the small amount of carboxylesterase 2 as well. So it gives you some idea of the importance, and obviously it has some other functions because there are endogenous substrates for carboxylesterase 1. And then if you look at in drug metabolism, so this is a survey from Matt Cerny, just looking at top 200 prescribed drugs in USA from, I believe it was 2005, 2016, with prodrugs excluded. And these are non-P450 enzymes. So no surprise, 45% UGTs, and then followed by some of the others such as salts. FMO, MAO are surprisingly small, and then carboxylesterase comes in there at 7%. So again, carboxylesterase may be overlooked in some cases, but it is important. You've been looking at these top 200 prescribed drugs. But if you also look at prodrugs, so these are established prodrugs that have been around for a while, just lumping them into hydrolyzes. So again, these could be any number of hydrolyzes. They don't have to be serine hydrolyzes. Could include carboxylesterase. You see quite a few, 78%. And then demethylases, obviously that's going to be mostly P450, coming in at 22%. And then if you look at the FDA-approved prodrugs from that time period, hydrolyzes, again, very high percentage, 75%. And then kinases now are starting to become more important for prodrugs because of the anti-HIV drugs, other antivirals that require the kinases to activate them. So a little bit of a change, and these are from 2 different publications over 2 different time periods, and we're continuing to follow these trends over time. So when it comes to substrate specificity, I think these are rules of thumb. There can definitely be some overlap. But if you look at this is from a book chapter that I and Mashi have had the pleasure of revising a number of times over the years with Andrew Parkinson. It's in Casper and Duell's Toxicology. And so the latest edition, 2019, I think we actually worked on these figures together, but Belapril is a good example of a carboxylesterase 1 substrate. So what carboxylesterase 1 seems to prefer is a large acid. So here's the acidic moiety on this large compound. And the leaving group, after water is introduced, is just a small alcohol. Whereas carboxylesterase 2, on the other hand, is the exact opposite. You can see that with the example of procaine. Here you have the small acid and then the rather larger alcohol as a leaving group. And what's large, what's small can differ. And like I mentioned, there can be some overlap. But that's the general rule of thumb. Now, when it comes to inactivation of drugs, that's one of the main purposes or reasons why there are so many xenobiotic metabolizing enzymes is to take something that may be toxic and make it less toxic. So in this drug metabolism, the idea is to terminate this pharmacological effect. So 2 drugs, there's Focalin, which is simply the D-enantiomer methylphenidate, and then you have Ritalin, which is the racemic mixture. And it turns out in this case that the L-methylphenidate is actually a better carboxylesterase 1 substrate, which I think is partly why Focalin is a little bit better efficacy for ADHD but both of them are converted to the rhyllinic acid, which is the inactive species. And so if you have differences there is one polymorphism I'll talk about with carboxylesterase. You can see some differences in the efficacy of focalin, for instance. You might have a longer half-life. But interestingly, and this is true for all carboxylesterase substrates, when ethanol is present, it actually prefers ethanol instead of water. And so because of that, it inhibits carboxylesterase 1 to some extent, but you can also then end up with transesterification. So in this case, you'll see this transesterification of, you get EL-ethylphenidate. In some cases, those metabolites can actually be more toxic. So one example that's commonly given is people who, hopefully not very often, are using both cocaine and alcohol at the same time. And the transesterification of cocaine can actually be more toxic, especially in the presence of alcohol. So with activation of drugs, so again, I mentioned the strategy for prodrugs, and that's been commonly used for a number of decades. But if you look at dabigatran, this is a very interesting example. One I think we've all become familiar, most of us have become familiar with over the years as a PGP substrate and marker. But if you look at the fact that it has two sites on it where carboxylesterase 2 and carboxylesterase 1 act on it. So again, from the rule of thumb, this is the larger leaving group as the alcohol. So carboxylesterase 2 catalyzes that first step to this M2 metabolite, which still has the ester on the end. And then carboxylesterase 1 will then catalyze that, and then you have the small alcohol leaving group to end up with the active moiety. So this is a case where they're actually using both enzymes to get to the active species in this case. Now with irinotecan, there is again overlap, but it's mainly catalyzed by carboxylesterase 2. And what happens here is you have this large leaving group, and you end up with SN38, which is known to be toxic. And I'll talk a little bit more about that. You get the gastrointestinal toxicity associated with that. In some cases, some fatal outcomes have occurred because of that. So just some examples, and this is a striking example. If you look at the ACE inhibitors, so many of these take a prodrug strategy, and what they're doing is they're exploiting the fact that carboxylesterase 1 can catalyze that leaving group, and you end up with the active species. In fact, there is a polymorphism that has been described, I think, for trindolapryl in particular, that can actually be substrate dependent. So depending on which ACE inhibitor or which other enzyme, that polymorphism can decrease the efficacy of the drug, and in this case, if you're doing it for blood pressure, can have a pharmacodynamic effect where you don't have the decrease in blood pressure that you would expect. But also looking at antivirals, so oseltamivir, it's marketed in the US and I think other countries as Tamiflu. That's the one where, within 24 hours of flu symptoms coming on, you would take it, decreasing the symptoms. You can also take it prophylactically. I had a case personally of this where my wife and daughter were diagnosed with the flu. I hadn't gotten symptoms yet. They gave me the prophylactic dose of it. And I don't know, I kind of wonder if I'm one of the people with the polymorphism, because I felt terrible and I wish I had not taken oseltamivir and just had the flu, honestly. It was that bad. But then also some HCV antivirals are also using this as a prodrug. And you'll see in this table, and you'll get these slides later, of course, anything that has a footnote is a prodrug that requires activation. And then when you look at some of the CNS drugs, I mean, a lot of these are drugs of abuse like cocaine and heroin, but even methylphenidate, like I mentioned, it's inactivating it and leading to the termination of pharmacological effect. But then even flumazenil, so this is an interesting one. Flumazenil, for anyone who knows anyone that works in hospital pharmacies, it's a sentinel prescription. If you see a lot of prescriptions for flumazenil, it means that someone somewhere is prescribing way too much midazolam or some other benzodiazepine. In fact, the joke is that the brand name for midazolam is Versed, and they use flumazenil to reverse it, so they call it Reversed. Not the brand name, but that's how some pharmacists refer to it as. Also, these anti-cancer agents, I've mentioned irinotecan, but also capecitabine, and then clopidogrel. I'll talk a little bit more about that one, and dabigatran. And then with the organophosphates, so not just the chemical warfare agents, there have been a lot of studies in those in the 80s, and even some more recent ones. I'll cover those in a little bit. But occupationally, people that work in agriculture, they can be exposed to these. Until maybe 10 years ago, you could still buy some of these stronger organophosphate pesticides, and just spray them on your lawn. But they've found, obviously, it's not the carboxylesterase that leads to toxicity. It's the fact that they also inactivate acetylcholinesterase, and so occupational exposure there can be quite important. And you need to have carboxylesterases along to help deactivate those. The stronger organophosphates will actually irreversibly bind to those and cause a longer-term inactivation. But in addition to xenobiotics, there are also some endogenous compounds, and I'll talk a little bit about those in a minute. So carboxylesterase, 2 examples. I just have a shorter list here, but I've mentioned dapagatrin, also peroxywill. But you can see again all the even some of the sartans. So sort of the counterpart, if you will, to the ACE inhibitors. They're more direct angiotensin receptor blockers. Will use this prodrug strategy to have activation by carboxylesterase 2. And then anesthetic drugs like procaine that have been used for years where that terminates the effect. And then moving on, then I've mentioned prodrug activation. That's one where it's been used for decades. And then also some newer modalities. So this is kind of a more exciting area where you're looking at some of the lipid nanoparticles that can exploit carboxylesterases, even gene therapy, and I'll cover those in a minute. And then potentially as a therapeutic target. So with prodrugs, what you want is to have weak or no activity of parent drug, right? And then you want chemical stability across a broad range of pH because you don't want it to get into the stomach and then be degraded immediately. You want high aqueous solubility, and then good transcellular absorption, obviously. You want resistance to hydrolysis during that absorption phase, so you don't want that to occur until it actually ends up in either the liver or the gut, where it's hydrolyzed by carboxylesterase 1 or 2, and then rapid and almost complete conversion to yield high concentrations of the active moiety, so you get good absorption, good profile pharmacokinetically. So this is actually from a Moderna paper, and Moderna actually kind of made their name with lipid nanoparticles even prior to COVID, which, they use their lipid nanoparticle technology for that vaccine. And what they've been looking at is the combination of this ethanolamine head group and then 2 different lipid chains, and putting a primary ester at the C8 position and then having a secondary ester in the second tail. And they looked at a variety of these to see which ones would help release the payload, usually an mRNA in this case, from the nanoparticles. And it turned out that for whatever reason, this number 5 here was ideal for delivering the mRNA in a variety of different modalities. So here's a case of gene therapy. So this is interesting, where this company is using single guide RNA for CRISPR-Cas9. And what they've developed is what they call a carboxylesterase responsive nanoparticle. And I won't go too much into the technology about it, but suffice it to say what happens is that this is a large nanoparticle, and it's delivering this single guide mRNA. And this is for the CK9 inhibitors. So this is for people that have high, very, very high cholesterol, the ones that have the familial disorder that you really can't treat with just statins alone. And so this gene therapy, the way it would work is once it actually escapes the lysosome, so it doesn't go through the endosomal lysosomal trafficking pathway, and then it hits the carboxylesterase present in the endoplasmic reticulum, where the payload is released, and then you can have the gene therapy and transcription occur. So again, I think this is something that cell and gene therapy companies are looking at, companies that look at lipid nanoparticles. I mean, with gene therapy across the board, whether it's transient like a vaccine like the COVID vaccine or something like that, or more permanent like true gene therapy, there's a variety of different ways. Lipid nanoparticles are one of the biggest ones, along with viral, and then also looking at how to release the payload in a controlled manner. So that's essentially what this company has done. And then going back a few years, this is more of an academic pursuit, but looking at carboxylesterase 1 as potentially a therapeutic target. So in this case, because carboxylesterase 1 is responsible for hydrolysis of a number of endogenous compounds. So if you think of esters of triacylglycerol, even some of the cholesterol esters, you can see potential benefit for inhibiting carboxylesterase 1 and lowering cholesterol, triglycerides, and maybe even potentially leading to treatments for obesity or even atherosclerosis. So what this lab did is they developed a knockout for a global knockout of carboxylesterase 1, and then along with a LDLR receptor knockout. So to walk you through this a little bit, what happens is the single knockout, the LDLR receptor knockout, as expected, you would see very high levels of triglycerides and cholesterol. But in the double knockout, so you still lack the LDL receptor, now you've knocked out carboxylesterase 1, and you can see that triglycerides and cholesterol both decrease. And they even showed that the atherosclerotic root lesion is decreased in the double knockout as well. So it may be an area for future focus in terms of therapeutic areas. Again, if you inhibit carboxylesterase 1, you're not likely to have a lot of other deleterious effects except for some of these drugs that could cause drug-drug interactions, but it could be a therapeutic target more in the future. And then, interestingly, carboxylesterase 2, no endogenous substrates have been identified yet that I could find. But that's true of a lot of these drug metabolizing enzymes, right? They're not there just to metabolize drugs. I heard an analogy one time that the whole system of xenobiotic drug metabolizing enzymes are there to handle any chemicals that might come our way. It's kind of like the battle between plants producing chemicals, and over evolutionary time, then these enzymes developed to handle whatever came their way. And just like P450s, there's some of those P450s that are involved in cholesterol, also the hormone metabolism and catabolism. So I'm sure there's got to be endogenous substrates for carboxylesterase 2. We just haven't found them yet. So species differences, these are challenges, and this is probably the main one, but also age and disease-related changes, and then some polymorphisms, as I've alluded to already a few times. So the main problem with species differences is the fact, let me walk you through this a little bit. So again, we have two, mainly in the liver, carboxylesterase 1, and then followed by carboxylesterase 2, and then the intestine, you can say we essentially lack carboxylesterase 1 for all intents and purposes. And then in monkey, we also have two. They have two, and the problem is they also have carboxylesterase 1 in the intestines. So you'll see a difference there between non-human primates and humans with intestinal substrates of carboxylesterase 1. And then with dog, they don't have either in the intestine. So that's a difference that can come up in certain tox studies where you might have carboxylesterase 1 or 2 substrate, and it behaves differently in dog because of that lack in the intestine. And then with rats, they've got 10, mice have 11, and they're organized into these different varieties of carboxylesterase 1 and 2. But the main difference here is the fact that I alluded to earlier, that KDAL receptor, the sequence is different for carboxylesterase 1 isoforms. So they are actually secreted into the plasma. And so what that means is extrapolating metabolism of carboxylesterase 1 substrates from rodents to humans is actually quite difficult. And so just to provide an example, again, this is a study from the 80s, and surprisingly there's even some more recent work on this where they must have a specialized lab where they can actually work with SOMAN. Main toxicity there, of course, is inactivation of acetylcholinesterase. But if you look at the LD50s, because it's secreted, carboxylesterase 1 is secreted into the plasma in rodents, it acts as a scavenger for SOMAN. So the LD50s are quite a bit higher than they are for guinea pig and rabbit. Interestingly though, if you inhibit globally carboxylesterase 1 with this inhibitor, then you'll see that the LD50s actually drop down to about the same across species. So again, that species difference will come into play not only for organophosphates, but any other drug that might have metabolism by carboxylesterases. So another challenge is polymorphic expression. So there's really one loss of function SNP that's been identified as the major clinically relevant SNP, and it has a sort of codominant effect because it turns out that even heterozygotes will have a significant impact on the disposition of carboxylesterase 1 substrates. So taking oseltamivir as the example, this is why I wondered if maybe I'm a carrier of this gene. You get a 360% increase in the AUC of the parent, which means very little of it, at least in the time frame that is dosed, is being converted to the active moiety. So in those patients, then you obviously don't get the efficacy, you don't get the decrease in flu symptoms that you otherwise would, and you probably have some increased adverse events from the parent. And in a pharmacokinetic drug interaction study, so this is looking at racemic methylphenidate, so this is Ritalin, and in ethanol, one subject actually was discovered to be carrying both carboxylesterase, the SNP above, and another one, and had even more profound effects in the disposition of methylphenidate. And then the major allofrequency for carboxylesterase 1, looking across species, different ethnic groups, I'm back on the species differences, is about 3.7 down all the way to, so up to 4 and then down to 0. So going across white, Hispanic, and African American. Asians, at least in this paper, completely lack that polymorphism. So you would see some significant differences across different ethnic groups based on that polymorphism that's present. But because, this is major allofrequency, so to find one that's actually a homozygote, you might have to screen something like 1,000 different individuals to find one that is homozygous. But again, even the carriers will have a significant impact here. So this is something that has to be considered if you have a drug that is predominantly metabolized by carboxylesterase 1. So going back again to clopidogrel. So this is one of my favorite examples. I did quite a bit of work on this myself because one of the P450s that's involved in actually activating it to the active species is CYP2C19. And I looked at the fact that esomeprazole inactivates 2C19 and can cause the interaction that's been described for years between proton pump inhibitors, in particular esomeprazole, with clopidogrel, which is an antiplatelet drug. But at each step along the way, carboxylesterase is shunting off to an inactive metabolite. And so if you have this loss of function, then what you would expect is that you're going to have more of the active metabolite. And that active metabolite, this is how complex, this is what I would call an example of irrational drug design. This probably would not be developed today. The active metabolite is one of 7 different diastereomers, and it has to go through multiple steps to actually get there. And the fact that you have carboxylesterase 1 taking away from getting to the active metabolite along the way is really not the sort of thing you'd want in active practice. But it is what it is, it's still used, and you can see across 2 different studies, the active metabolite is increased anywhere from 60 to 67%, and in concordance with that pharmacodynamic effects, antiplatelets anywhere from 24 to 31% increase. So we've got both the pharmacokinetic and the pharmacodynamic evidence here across 2 different papers that SNP in carboxylesterase 1 can in fact affect clopidogrel's efficacy. And then when it comes to age-dependent effects, so these are data from a couple different papers. Also, Ron Hines, who's made his name for ontogeny of different drug metabolizing enzymes over the years. And if you look at the age-related impact, so this is, just to get your bearings, this is a log scale. But you can see from neonate to about age 1, you're starting to increase for both carboxylesterase 1 and 2 until you get to more adult levels. And so these studies looked at oseltamivir, metabolic stability. You can see that there's a large difference between adults and pediatrics. And so what this means for drugs like oseltamivir or potentially any other of the prodrugs is that you're going to have less efficacy in pediatrics. So this may come into play in special populations where you're looking at a prodrug. There are cases where pediatrics would need something like an ACE inhibitor in some cases, but in this case, oseltamivir, if you administer Tamiflu to a 2-year-old with the flu, you may actually have less efficacy in that case. And then also disease state. So we know that alcohol directly inhibits it, but also the long-term effects of alcohol, so liver disease. And this is just going across Child-Pugh A through C, so mild to severe cirrhosis. And you can see this is a proteomics approach again, and that with mild cirrhosis, you get about 50% decrease in carboxylesterase 1. There's really no change, at least in this study, with carboxylesterase 2. But then over the severity of liver disease, you see that incremental decrease up to 92% loss of protein of carboxylesterase 1 in severe cirrhotics. And so you can see that another study was performed, I think this was it was after the fact, it wasn't during the approval for dabigatran, but they showed that across different grades of liver disease, that you see then less effect of dabigatran and less of the active metabolite actually being formed. So some concurrence between what you would expect and then the PBPK modeling. So then I mentioned irinotecan earlier. So tissue distribution and UGT abundance. This is looking at abundance per milligram of S9 protein. And so in the intestine, a large amount of carboxylesterase 2. And then looking at just the abundance per gram of tissue, again, large amount in the intestine versus the liver, and then you can see the other tissues. And then abundance per organ. So again, carboxylesterase 2, there is still quite a high abundance in the liver, but also in the intestine. And since irinotecan systemic clearance is driven mainly by carboxylesterase 2, and yet you don't see hepatotoxicity. But we know the well-known intestinal toxicity that you see with irinotecan, and that has mainly to do with SN38, which again is that alcohol that's the leaving group in that case, that causes the toxicity. So this paper puts forward the idea that the toxicity observed is likely due to the fact that you have much higher concentrations of carboxylesterase 2 in the intestine than you do of UGT1A1. UGT1A1 would then glucuronidate that SN38 and decrease its toxicity. So that difference in protein then may explain that toxicity, why you don't see hepatotoxicity. So then moving on to regulation, I mentioned this before that these were mostly rodent studies, but you go across the regular players, so PXR, CAR, but also PPAR alpha, HNF alpha, and even NRF2. And in fact, a few studies have actually showed induction of carboxylesterase 2 in human tumor cells by NRF2, and this could be more of an adaptive response. But then also PXR agonists, so rifampin, you can see moderate induction of both carboxylesterase 1 and 2 in hepatocytes. And again, don't forget there is carboxylesterase 2 in hepatocytes. I think it's easy to think of carboxylesterase 1 as being the hepatocyte carboxylesterase. And then dexamethasone and phenobarbital also going through different pathways can cause some moderate induction of both carboxylesterase 1 and 2. And then inflammatory cytokines. So this is known for a lot of different P450s that when you have inflammatory cytokines, you can see suppression. This holds true also with carboxylesterases, apparently. This study showed, even though it looks minimal, it is significant, that when you're in the presence of IL-6, you get significant suppression of both carboxylesterase 1 and 2. And so that may have an effect especially if you're in a disease population that has high levels of IL-6, for instance. So it's a little bit of a stretch to look for some of these clinically relevant drug-drug interaction studies with carboxylesterase 1. And I think that's partly because of a lack of a need to do a dedicated clinical DDI study with carboxylesterase 1 and 2. So this is a little bit academic. So they're using alcohol here to inhibit. And what they show is it significantly inhibits oseltamivir in vitro, and then clinically increased oseltamivir AUC by 27%. So it's just above that 1.25-fold increase where we would consider it to be a clinically significant drug-drug interaction, but it does have an effect. And I kind of thought this is a little bit less of a practical study because, do you need to put on the label that, you shouldn't drink alcohol with Tamiflu? I think if you're at home really sick enough with the flu to take Tamiflu in the first place, probably shouldn't be drinking alcohol either. So I hope not many people are drinking alcohol while they're sick with the flu. But when you look at aspirin, on the other hand, so this is carboxylesterase 2, alcohol doesn't actually affect its disposition at all. So a little bit of a difference between the effect of alcohol on carboxylesterase 1 and 2. And then if you look at drug-drug interaction potential, I don't have the clinical correlate here because in many cases it just doesn't exist. There wasn't a clinical study performed, but you can see that a lot of these compounds that are used widely will have very low Ki values towards carboxylesterase. And so for instance, fenofibrate, which is actually dosed at a relatively high dose, has submicromolar 0.04 micromolar Ki. Even simvastatin for both carboxylesterases, we're looking at submicromolar Ki values. So there's a potential for inhibition there. And again, it could be that either the interaction wasn't noticed, but you can imagine that if you give these drugs along with an ACE inhibitor, you might actually see decreased effect. Again, there may be some case studies in the literature, but I have not seen any dedicated clinical DDI studies there. And then in addition to that, I mentioned organophosphates. So they're important for occupational exposure in agriculture. And what happens is they do irreversibly inhibit these enzymes, but there is a recovery period, so they can actually come back off, but it's pretty slow. And when you get to the chemical warfare agents, they've been in the news in recent years, mainly with some of the assassination attempts that have occurred. Novichok, I think, is one of them that uses some of those organophosphates as nerve agents inhibiting acetylcholinesterase. They don't have the time for that recovery period to occur, so that's why they're more toxic than the typical pesticides that are used. So when it comes to de-risking, I'll talk a little bit about metabolism. So this is the victim potential, or now as they're going to call it, the what do they call it? The precipitant potential. And then carboxylesterase inhibition. So this is the perpetrator potential, and then induction and suppression. So to look at victim potential, first look at the structure, right? If you've got one of these, it's an ester, thioester, carbamate, even amides can be catalyzed by carboxylesterase 1. Take a look at it, decide, okay, maybe we need to take a look at this, and then look at in vitro metabolic stability. And because there are many other hydrolyzes that could potentially hydrolyze the catalysis, you would want to look at plasma first, because if you see a difference in plasma stability versus human liver microsomes or S9, that might tell you that there's something beyond carboxylesterases that's hydrolyzing it as well. And then look in liver versus human intestinal microsomes, so you can tell if there's a difference between carboxylesterase 1 and 2. And then also looking across species, because as I mentioned, the species differences with carboxylesterases can be quite significant. Next thing would be to look at the in vitro versus in vivo profile. So if there's differences there, that might tell you, again, across tox species, that you have a species difference. And so reaction phenotyping, then after that point, you've narrowed it down to carboxylesterases, becomes quite simple. You would look at the recombinant enzymes which are available from us, and then some chemical inhibitors. There's some pretty good selective inhibitors for both of these. And then when it comes to perpetrator potential, so now we're looking at just simply inhibition. Pretty easy, simple in vitro test systems, human liver and intestinal microsomes with well-known probe substrates, and just run IC50 values. For instance, you go on the Ki values if you really wanted to. And then for induction, this is easy enough to do by just getting the primer for the mRNA and looking for induction in hepatocytes. So that's what I've got. I think hopefully at the end of this you'll appreciate that these enzymes are maybe more important than they've been given in the past in terms of not only metabolism but drug-drug interactions, and that there are actually some opportunities. So some of these newer modalities, like I mentioned, companies are really starting to take a look at is there a way to exploit not only the old prodrug strategy, but the strategy for delivering mRNAs or gene therapies, for instance. And then the drug-drug interactions shouldn't be overlooked, especially in the cases of prodrugs. So if you inhibit to such an extent that you're not getting the beneficial effect of these antivirals or ACE inhibitors, then that could be a problem. And then also the polymorphisms. So those seem to be even more of an impact than drug-drug interactions. So if you have someone that has the SNP for the carboxylesterase 1, you could have a dramatic effect that potentially is even substrate dependent for ACE inhibitors, for instance. And the methodologies I mentioned, I just covered at a high level, but certainly we have more detail for you if anyone's interested in those studies. So with that, thanks for everyone's attention. I'm happy to take any questions.